And they have to involve a change in the frequency of genes, right? So the gene pool is what's changing over time, because the individual genes in a population are going to change from generation to generation. And so the gene pool is going to change over time, over generations. So this is what's happening, right? Because changes in the gene pool are going to result in changes in phenotypes, the things that we see, right? So again, evolutionary change can lead to adaptations and must involve changes in the frequency of individual genes in the population from generation to generation. So we have a change in the gene pool over time. Okay, that's evolution. Evolutionary changes, one of the mechanisms they can come about from is natural selection, right? We talked about there's other processes that can cause evolution as well, cause change, like mutations, genetic drift. One we didn't mention last time that we could add to the discussion is immigration and emigration. As individuals move in and out of populations, they take genes away and carry new genes in, right? So those are other ways that gene pools can change over time. But the one that we're talking about is natural selection, because it's probably the only one that leads to what we would call adaptations. So the process of natural selection then is the end result of the process of ecology in action. So we've been, the first lecture, we talked about a definition of ecology. We talked about a few ways to define that term. So you can think of natural selection then as the end result of this process of ecology in action. That is the environment shaping the organisms that live there, right? Because the environment is the selective force that works under natural selection. Okay, so the environment that the organisms inhabit, so the animals in our case, because it's of course an animal ecology, so the environment that those animals inhabit is what's going to shape the changes that happen, right? It's going to shape the evolution that occurs, because it's that environment that's going to be making those selective forces that cull out some individuals and favor others so that they can survive and reproduce. So if you remember, our definition of ecology included these terms: distribution, abundance, those types of terms, right? So the present distribution, like on a geographic scale, and the abundance, how many there are, and also the biodiversity of animals, so what different species are in a given place, and plants, those are going to be set by evolutionary processes that happened in the past, impinging on the environment of the present, right? So there's selective forces have been acting. And so what we see now as those organisms and what we would call the adaptations that they have, those things have been shaped by the processes that occurred over time. And then we see the result now, those adaptations. Okay. So the word adaptation, it's one of those terms in ecology that's had lots of different definitions and meanings attributed to it. So I've got a few of them listed here, and we'll talk about those. And they all sort of overlap, but this is different ways you might have seen adaptation sort of defined in an ecological context. So the first one is any behavioral, so behaviors, morphological, so physical features, or physiological traits, okay, that are assumed to be the result of natural selection acting. And so this is the way we defined it last time when we were talking about natural selection. So that results in adaptation. So these behavioral, morphological, physiological traits that have resulted from selection acting. Another definition, and again, they sort of overlap, any physiological or morphological feature or form of behavior used to explain the ability of an organism to live where it does. So that's kind of a just more wordy way to say explain the ability of an organism to be in that environment. It's the way the environment has been acting on those traits, okay? And then the third one, an inherited characteristic. So this is an important one to note, right? An inherited characteristic that enhances an organism, an animal's ability to survive and reproduce in that given environment. And if you remember, an individual's relative ability to survive and reproduce is its fitness, right? And this is where that term of Darwinian fitness comes into play, right? So, and also remember those four tenets of natural selection that we talked about last time, is that those traits are heritable, right? Okay. The fourth one is a bit different, and it's not, I would say, is not quite a true definition of adaptation. So number 4 says a change in physical, physiological, or behavioral traits that result from current environmental pressure. So I've got the asterisk there because this is not really adaptation in the evolutionary sense. It's kind of more acclimation, right? It's something, it's about an animal adapting or adjusting to a change in temperature, for example, right then and there. So it's not it's about the current environment. So I would say this is more in a sense talking about acclimation, and it's not so much an evolutionary process as it is a more current process. And so later in today's lecture, we're going to be talking about ultimate versus proximate factors, and this is going to come into play. This fourth definition, I would say, is more talking about proximate factors, which have to do with how an individual responds and decodes its genetic information, versus an ultimate explanation, which has to do with evolutionary processes. So again, this fourth one, I would put a caveat in there that I would not use this as a definition of adaptation in the evolutionary sense of the word. Okay, so the adaptation of an organism to its environment is exhibited by its ability to function between some range of environmental conditions, right? There's going to be a range. There's going to be a lower end and a higher end to what an animal can operate under. So organisms function, animals function between some upper and lower limits of a range of environmental conditions. They can't operate everywhere, right? Like there's a range. And so we're going to talk about tolerance curves and the law of tolerance. But first, we need to talk about a few things that lead to that idea. So there's been, over time, ecologists and biologists have been thinking about these things that ultimately resulted in this idea of tolerance curves. So we start with this idea of the law of the minimum. So this is about the role of the limitations in the response of an organism to its environment. And this was first recognized a long time ago in 1840 by actually an organic chemist, so Justus von Liebig. So he was an organic chemist, and he noted, so this idea comes from plants. In fact, so does the law of limiting factors. Both of these ideas. So von Liebig, he noted that plant growth responds to the nutrient that is most limited, the one that there's the least of, the most limiting resource. So if one of such mineral, like a mineral substance, for example, right, is present in minimal quantities only, then the growth of the plant's going to be minimal, right? That one nutrient that's the limited resource, that's going to dictate what happens, right? So this idea of that variable that's the most limiting is what what an organism responds to. This became known as the law of the minimum, that minimum thing, right? And so there's some restrictions on how you can apply this law of the minimum. So it applies only under like steady state conditions when everything else is available in excess of what the plant in this case needs, right? So it only applies. So there's these restrictions to this law, and that it only applies under steady state conditions when all the other resources are available in excess of what the plant in this example needs. So if the quantity of the limiting resource, say it increases, so say that there was some single mineral in the soil that was the limiting resource. If that mineral that increases in availability, it no longer is limiting, right? It no longer is that limiting factor. But another resource, some other nutrient in the soil perhaps, then is the one that becomes the limiting resource and will impact plant growth and survival, etc. So this law of the minimum applies when we only have this steady state condition, when everything else is all the other resources are available in excess, like the plant is not limited. Okay. The second law to mention here is the law of limiting factors, which sounds sort of similar in the title, right? But this is subtly different. It's more along the lines of too much of a good thing can also be bad, right? So for example, too little light, right, is detrimental. So again, this comes from plant ideas. So too little light may be detrimental, but too much light is also harmful. If you ever have grown, a seedling on your windowsill, like a tomato seedling or something, and you put it there and it's just too much in the direct light, you end up with this really long, spindly, can't even stand up on its own type of plant, right? Because too much light is also harmful. And so this idea was advanced by F.F. Blackman in 1905. And they pointed out that a maximum quantity of a resource tolerated by an organism can also limit its response, right? So too much of a good thing can be bad. So for example, the response might be growth in my example of a tomato plant, right? So again, this idea is that a maximum quantity of a resource that's tolerated by an organism could limit the response, the growth response, or whatever it might be that the response variable is that we're talking about. So this idea became known as the law of limiting factors. Okay. And so the law of the minimum and the law of limiting factors, right? They're both emphasizing environmental conditions. The law of the minimum is emphasizing resources. And in our example, we talked about mineral resources in the soil for a plant. And the law of limiting factors also talks about resources. And we gave the example of light. Too much light is bad, just like too little light is bad. So both of those laws come out of this idea about environmental conditions. And so, the reproduction and growth and functioning of individual animals is going to depend on the amount of the essential environmental requirements present for them, right, in some minimal quantities during especially the most critical season of the year, right? So there's going to be, that reproduction, the growth, the functioning of individual animals is going to depend on the amount of the essential environmental requirements that are present for them during the most critical times of the year, right? So, and think about this in terms of, we can think about birds, for example. So the availability of food is critical, not during winter, which you might sort of think of first off, but it's actually in the shoulder season, for example, between winter and spring, the beginning of spring. Food resources are really scarce at that point, right? But the bird is already ramping up its behavior to get ready for mating and its reproductive season. So birds are pretty active in that early spring period, but the resources for them aren't quite available yet. So that's a really challenging time of the year. So again, it's this idea that the reproduction in that particular case of those birds is dependent on the amount of the essential environmental requirements present for them that are in minimal quantities at that time of year, right? So, seed or whatever they might need to eat. The plants haven't produced seeds yet. So that's a challenging time, for example, for birds. And so this leads to this idea that organisms animals live within some range of too much and too little, right? The limits of their tolerance of what they can handle. And so Shelford in 1913 incorporated these ideas together, this environmental limitations on an organism's ability to reproduce, to survive, to grow, into this concept known as the law of tolerance. Okay, so again, bringing in these ideas of the range of too much and too little, and the limits of tolerance of what an organism can handle. So it's about environmental limitations that impinge on an organism's ability to survive, grow, reproduce. Okay, and so this comes to the law of tolerance. And so it's illustrated by this bell-shaped curve, right? So we've got on the x-axis, we have some environmental condition, could be temperature, could be humidity, could be salinity, could be whatever. And on the y-axis of the graph, we have the response of the species. And this could also be different things. Like it's going to have to do with fitness, right? Because we're describing this in terms of fitness. So it could be represented as population abundance or any number of things, but the response of the organism, whichever response variable you're measuring, reproductive output, those types of variables could be put on that y-axis. Okay, so again, the x-axis is some gradient of environmental conditions from low to high, and the y-axis is the response of the species that we're talking about. Okay, and then you come to this curve, this bell-shaped curve, and the example here is a bird that we're using. So the middle part of the curve, where it's the tallest, here's where we have the greatest fitness, right? This is where the animal is within this range of environmental conditions, from this low end to this high end. The animal is super happy, right? It's able to survive and grow and reproduce. And so if your response variable here is population abundance, that's within that range is when population abundance is going to be greatest, it's going to be highest, right? So this is the range of greatest fitness. But then as you move towards the downward swings of the curve on both sides, now in this range of environmental conditions, the animal is a little more challenged, in this case, the bird. So it's able to grow, it can put some energy into growing, but it's not able to also reproduce. It's just, keeping itself on a growth trajectory, but it doesn't, it's too compromised to be able to reproduce as well. And then as we move out even further into the shoulders, and you can see the bird here in this posture, now, at these sort of tail end extremes of those environmental conditions, say it's temperature, this cold extreme, this hot extreme, the animal is just surviving. It's just holding on, okay? It can't grow, it can't reproduce. Really, all it can manage to do at that point is survive. Okay, so you can think about maybe, a winter condition. The animal can survive, but it's not growing and reproducing, right? And then you get to this critical minimum point and this critical maximum point where the organism is now absent or dead, right? It can't survive. That is the zone of intolerance. We have a lower limit of tolerance, a lower critical temperature, for example, and an upper limit to tolerance, or maybe in this case, if it's temperature, an upper critical temperature. So after that, the animal cannot tolerate it. It's dead. The organism is absent. Okay. So that's an example. So here's another example that's just showing some sort of different ways of looking at it. So again, we have an environmental gradient on the x-axis, and now it's abundance. The response variable that we're talking about in this case is population abundance, right? And so here we have organisms in greatest abundance. And you can think about that, right? When you think about fitness, basically population size is kind of a surrogate for fitness, right? Because if the organisms are fit, the abundance of the population is going to increase because they're going to successfully survive and reproduce, right? So just another way to look at it. Okay, so again, we have the bell-shaped curve, right? We have the highest abundance of that in the population of that organisms, but we get to these edges where we have this physiological stress. The animal becomes uncommon in that type of environmental gradient. And then, of course, the zone of intolerance where the organism is now absent. So this graphic doesn't have the sort of two stages that the previous graphic had that showed where the organism could be maybe just growing versus where the organism is just really quiescent and just surviving. Okay, so just another example to look at it. We can think about it across here, for example, as a gradient of pH maybe, right? So we have acidic conditions here, and we have neutral conditions in the middle, and basic conditions at this end of the gradient. Okay, so here's another example, and here we're talking about specifically salinity tolerance in fishes, and also talking about tolerance ranges, which can be either broad or narrow. Oh, I see a chat. I'll just go in there real quick. When talking about, it's a private comment, but still, okay. When talking about the curve and the area of growth with no reproductive activity, does that mean the animal won't even try reproduction, or is it that they try and they're just not very successful? So I think that what that means is they don't even try. They don't have the energy to try. They're just so later on in the course, we'll talk about energy budgets. And for those of you that had herpetology, we talked about that a little bit as well. So they don't even have the energy to invest in reproduction. So they in most cases, I would suggest that they don't even try. Okay. All right. So back to the sort of tolerance curves that can be either narrow or broad. So the tolerance curves are broad for lots of animals. So they're able to, what that means, right, is they're able to exist within a wide range of values along that environmental gradient. So a particular environmental gradient of maybe, in this case, it's salinity, but it might be temperature, humidity, whatever. But then other animals have a really narrow range of tolerance curve, and sometimes particular stages of the life cycle have particularly narrow tolerances. So when you think about the life stages of an animal, there might be an egg stage, a hatchling stage, a neonate stage, juvenile, and then adult. And those different stages might actually have variation in their tolerance. They might be have a narrow tolerance at some life stages and a broader tolerance at other life stages, okay? But regardless, that whichever one has the narrow tolerance, right, that's going to dictate where that organism can live. Okay, so if we have some root words here that help us to define these ranges, right? So we have steno as a root word. So this means the organism can maintain homeostasis, and we'll come back to what that is in a little bit, but I think a lot of you probably know already. But an organism, so steno is an organism, an animal that maintains homeostasis only within a narrow range. Okay, so it's this very narrow range. It reaches a taller height, the curve here, but it's within a narrow range. So in this particular example, we have a salinity gradient, and we have a narrow range. So we have a stenohaline, right? So stenohaline fish. And so that's, for example, a coral reef fish like this guy, who can only handle a narrow range of salinities that are on that coral reef, right? So again, an organism can maintain its homeostasis only within a very narrow range of environmental conditions. So again, a stenohaline fish, like a coral reef species, can tolerate a narrow range of salinities. If we think about an animal's thermal tolerance, we might say stenothermic. So stenohaline, the haline part, is telling us that that's about salinity. So if we said stenothermic, that's telling us about temperature tolerance, right? So we could say a stenothermic reptile, maybe a snake that lives in an equatorial region, has a narrow range of temperatures, right? So stenohaline fish, a coral reef dwelling fish that just can handle a narrow range of water salinities and marine conditions. And then if we said a stenothermic snake, living at the equator, can only handle a narrow range of temperatures in its environment. Okay, the other part of the flip side of that, the opposite, is Eury. Eury, haline or eurythermic. Eury, that root word, means that the animal can maintain homeostasis across a broad range of conditions. So this lower curve, this example of a Eury haline fish would be a fish that could tolerate a wide range of salinities. So this might be a fish species that lives in an estuarine type situation where fresh water inundates out and then salt water mixes in. And so there's quite a wide range of salinities in the water conditions. So again, a Eury haline fish, such as the species that lives in an estuary, can tolerate a broad range of water salinities. And then back to a temperature example with reptiles. So a eurythermic reptile, you can think about temperate zone snakes. So snakes that live here in Sudbury, like garter snakes or red belly snakes, those have a wide range of temperature tolerances that they can handle. Those eurythermic snakes that live in temperate zones, like here in Sudbury, that will then be able to handle a wide range of temperature conditions. Okay, just to go back for a second on this definition, which we'll come back to. So what is homeostasis? Homeo tells you same, right? So what does somebody want to put in the chat a definition of homeostasis? Give it a try. Same standing. Let's see. The maintenance of internal conditions. Right. So both Cameron and Noah have said the maintenance of organisms' internal equilibrium, but relative to what? So keeping a constant internal environment relative to its surroundings, exactly. So the external environment can be fluctuating, and it often is, right? So homeostasis is an ability of an organism to maintain a constant internal environment regardless of what's happening on the outside of its body. So this has to do with osmoregulation and pH balance and all that kind of stuff. Great, awesome. Okay, and we will come back to that. We're going to talk about homeostasis a little bit more. Okay. So the other thing about tolerance curves is they're not absolutely fixed. They can move around. They can shift. So the range of tolerances is not fixed for all animals. So as seasons change, as conditions change, individuals might acclimate and shift their tolerance curves back and forth across that gradient of environmental conditions on the x-axis, right? So here's the tolerance response and a gradient. So you have, this, but then you can have the gradient, the tolerance curve shifting lower on the gradient or the tolerance curve shifting higher along the gradient. So for example, if you have a fish, right, that lives in a pond, and in that pond, the water temperature is going to change seasonally. So in the summer, the water temperature is going to be pretty high, and in winter, the water temperature is going to be pretty cold. So as the water is warming up in the spring, the fish's tolerance for warmer temperatures gradually increases, right? So its ability to tolerate those warmer temperatures is shifting to be able to handle those warmer temperatures. So at the same time, its tolerance for lower temperatures is going to be diminished. The whole thing is shifting, right? So as it's warming up in the spring, as that water is warming up in the pond, the fish's ability to tolerate those warming temperatures gets better and better, but at the expense of not being able to handle the cooler temperatures anymore. So then you flip that in the fall, when the water temperatures start to cool down, the fish's tolerance curve also shifts down. So its ability to tolerate those colder temperatures gets better, but at the expense of its tolerance of warmer temperatures. So for an animal that does this shifting, what it means is that if you took a cold acclimated fish and stuck it in summer temperatures, that could be lethal because it's not having its tolerance curve, including those alternate temperatures at that point, right? So similar, if you took a fish in the summer who's acclimated to those warm conditions and suddenly put it in the cold conditions, it wouldn't be able to handle it. But if it was winter, it could handle it, right? So just this idea that there's shifting, the animal can't tolerate this range from the low extreme to the high extreme all the time. The curve is narrower than that and it's shifting across that gradient. So I hope that makes sense. Okay, so although there is this shifting, right, this tolerance ranges can shift with the season, all of the shifting does take place within a finite limit, though. There are physiological limits, right? These are dictated by ultimate factors. And again, we're going to come back to that idea of ultimate versus proximate. But ultimate means the evolutionary, right? What's been shaped by natural selection over time. So there's going to be physiological limits that dictate how far that shifting can happen. Okay, so it's this plastic response, right? It means it changes. So there is some plasticity in this short-term shifting of these tolerance curves given different environmental conditions. And this is called acclimatization, right? This changing, this response, this acclimatization, this ability to shift around. But again, this lower limit of the shift and this upper limit of the shift, those are going to be fixed by ultimate factors, right? Evolutionary factors. Okay, so the law of tolerance explains a lot about local and geographic distributions and abundances of species, right? Again, that's back to those definitions that we talked about in the first lecture that have to do with ecology. And most species are restricted by the conditions, right, beyond their tolerance limits, right? They're not going to be there. We saw that in the curves, the absence of the organism once you get below the lower limit and above the upper limit, right? And most species are restricted, and I alluded to this already, they're restricted by the most sensitive stage of their life cycle, the most sensitive life history stage. And we'll come back to that a little bit too later in the lecture about life histories. But the most sensitive stage of their life cycle. So I said about eggs and hatchlings and neonates and juveniles and adults. It might be that one of those life stages is the one that's most sensitive. And so that's what's going to dictate the distribution of that organism in the environment. So an animal might have a wide range of tolerance for some part of its life cycle, like the adults maybe, but a really narrow range for another part, like maybe the hatchlings can only handle a very narrow range. And so that's what the hatchlings can handle is what's going to dictate the distribution. And so you can think about that. Little tiny animals have a really high surface area to volume ratio. So if it's an animal that is living in a humid condition, it needs to be in a humid condition. All that surface area to volume means that hatchling is going to lose a lot of water to the environment. So it might be that the humidity of the environment for that hatchling stage is the key to determining the distribution of that animal as a whole. I hope that made sense. So I have another example about snapping turtles, for example. The adult stage of snapping turtles, those great big beautiful adult snapping turtles, they can tolerate a range of temperatures from just above freezing, like plus 0.5 degrees Celsius, all the way up to like 35 degrees Celsius or even more. So, about a 35 degrees Celsius range of tolerances that they can handle. Now, it doesn't mean that they are perfectly, at the bell shape of that curve for the whole thing, but they can tolerate that range. But, like, for example, they might not be feeding and digesting and everything through that range. During hibernation, they're obviously not going to be doing that. They're in the just survival stage at that point. But if you think about the egg stage for snapping turtles, they have a much narrower range. So the range for egg survival, and if you go further and talk about embryogenesis actually happening, it's only about 20 to 35 degrees Celsius, a much narrower range than what the adults can tolerate. So in fact, for snapping turtles and other freshwater turtles in general, most scientists believe that what limits their northern range extent is those temperature conditions available for egg incubation. So adults, they can get into places to hibernate, for example, and move around to find what they need temperature-wise. But because of this life stage, that's the limiting life stage, most turtle biologists think that it's the incubation conditions for eggs that dictates how far north turtles can be distributed in the northern hemisphere. Okay, so these variations in tolerance are relating directly to differences in like the biochemical, the physiological, and morphological and behavioral characteristics, all those things that we've talked about, right? So these variations relate directly to these things, these biochemical, physiological, morphological, and behavioral characteristics of the animal. So the set of characteristics that enables an animal to maximize its fitness under one set of environmental conditions is going to limit its ability to do as well in another set of conditions, right? And so it's not going to be able to maximize its fitness across all conditions. It can't be a jack of all trades, so to speak, right? So it's going to be better at some things, better at some conditions than other. And so this idea implies what we call a trade-off, right? The characteristics that enable an animal to maximize fitness under one set of environmental conditions are going to limit its ability to maximize fitness under other environmental conditions. Okay, so there's a trade-off here. So this is a really important concept when we talk about animal life histories, and I will come to this later in the lecture. But this idea of life histories, life histories are characteristics that have to do with survival and reproduction, or fitness characteristics. And so a little bit later in the lecture, we're going to talk about clutch size in birds as an example of a life history trait. Okay, so again, as environmental conditions change, the set of characteristics, that is the adaptations, that allow an animal to survive and grow and reproduce are going to vary, right? So over the next few lectures, we're going to be talking about how animals are adapted to these various features of their physical environments. Okay, so back to that idea of homeostasis, to talk a little bit about that again. So when faced with a changing environment, animals need to maintain a pretty constant internal environment, right? And that's important because metabolism and other biochemical processes can be performed most optimally within a certain range of conditions, right? When it's too cold, metabolism slows down a lot. Enzymatic reactions go too slowly, etc. So when that environment is changing, animals have to still maintain a pretty constant internal environment regardless of what's happening around them. So they have to have some way to regulate stuff. They have to have a way to regulate body temperature, the water balance in their body, the pH, how much salts are in their fluids and tissues relative to what's in the external environment. And so again, this maintenance of a relatively constant internal environment relative to a variable external environment, that is homeostasis, okay? Maintenance of a relatively constant internal environment, even though the external environment is fluctuating. And so it requires that the animal has some means of regulating these things and detecting changes, right? So homeostasis involves a flow of external environmental information, right? So there's information that the organism is noticing. And so there's a system with homeostatic mechanisms. So these are these feedback systems that we're going to talk about. So again, there has to be some way of detecting and then regulating these different attributes of the internal body environment. And so this leads to this idea of feedback systems. Okay, so we're going to talk about negative feedback, positive feedback here. So, we're surrounded by chemical and mechanical homeostatic devices, right? For example, the thermostat in your house, right? So the thermostat for your heating system. If we want the room temperature to be 20 degrees Celsius, we set the thermostat for that temperature of 20 degrees Celsius, right? And when the room temperature goes below 20 degrees, there's a temperature sensitive device, right, that's detecting that in your house. And when it detects the temperature has gone below 20 degrees Celsius, below that set point that you've told it, then the thermostat is going to trip the switch of your furnace to turn on your furnace, right? So the thermostat's going to detect that and it's going to say, hey, turn on the furnace. We need to get back to that set point. And when the room temperature reaches the set point of 20 degrees Celsius, then the temperature sensitive device in the thermostat is going to say, okay, we're here, and it shuts off the furnace, right? And so you have this, there might be little fluctuations around it, but for the most part, that's what keeps the temperature of your house at that set point of 20 degrees Celsius. And so this type of response, when we have this halting or reversing of the direction away from and then returning it to that set point, that's called negative feedback, okay? So halting or reversing the movement or change away from, and then returning it to that set point. Okay, that's a negative feedback. Now, if that thermostat fails to function properly, that device in your house is not doing what it's supposed to do, and it doesn't shut off the furnace once the temperature gets to 20 degrees Celsius, then the room temperature is going to keep going up and up and up and up, right, until the furnace overheats and the whole thing fails, and you have to call Castle Electric and Plumbing to come and fix it. So this continued movement away from that set point, that's called positive feedback. So don't be confused with the word positive meaning a good thing in this case. In this case, positive feedback is actually a bad thing in a biological context. Okay, so again, it means that the room temperature in the case of the set point is going to continuously increase until the furnace overheats. And so this continued movement away from the set point is resulting from this positive feedback. So the measure of this feedback is directly related to the input. Basically, there's an enhancement of the effect by its own influence. So an example of this is cancer cells. Cancer cells are cells that have gone out of control, reproducing too fast. And so then they produce more cells that reproduce too fast. They produce more cells that reproduce too fast, right? So that's a positive feedback. The enhancement of an effect by its own influence. That is increased cell reproduction in the case of cancer, right? Okay. Another example is heat stroke and geometric population growth, which is a population growth model we'll talk about a little bit later. So again, despite the name with the word positive in there, it's actually not a good thing in a biological context. Okay, another definition here to mention are homeostatic plateaus. So a key difference between the thermostat in your house and animals, right, is that set points aren't firmly fixed for animals. Animals have a sort of, we talked about a limited range of tolerances, and that's bracketing what we would call a homeostatic plateau, right? This sort of range, there's a lower set point and an upper set point. So really there's kind of a range of set points that animals operate in, not like that single set point that you tell your thermostat to operate with. So it's sort of a range of set points. And so homeostatic systems work within that minimum and maximum value by using negative feedback, right, to regulate the activity around that range of set points. And again, if the system deviates from a set point, then the negative feedback response is going to inhibit further movement away from that set point and try to bring it back to the set point. If the system fails, of course, then positive feedback kicks in and drives the response away from the set point. That means something is wrong, right? Okay. So for animals, this, unlike humans, we are animals, but in our houses, we push a button to get a set point. But for animals, the control of this homeostasis is driven by physiological and behavioral aspects. So homeostasis is both physiological and behavioral. And again, those set points are not firmly fixed. There's some variation. Okay. So I posted this paper. So this is, again, Cameron asked for some invertebrate examples of things. So I added this paper because I thought it was really interesting because, again, it talks about various things that we are covering in this lecture and gives a neat example. So I posted the paper on D2L for you, and I have a summary of the things that I think are important to this story here. So it's about pH tolerances and the regulatory abilities, right? That set point, ability to keep set points in freshwater and euryhaline mosquito larvae. So freshwater is pretty clear, right? It's not salt water. So euryhaline. So what does that mean right away? What does Eury mean from what you just learned about tolerance curves? Somebody in the chat, tell me what euryhaline means. Broad, right? It can, exactly, excellent, guys. So a broad range of tolerances of salinity in this particular case, right? Haline means salinity. Awesome, good job. Right, so here we're talking about pH tolerances, which is something that can be on that tolerance curve on the x-axis. And we're talking about homeostatic mechanisms. We're talking about regulatory abilities. We're talking about two mosquito larvae that live in different environmental conditions, right? We have freshwater mosquito larvae and euryhaline, that is mosquito larvae that can handle a wide range of salt conditions, right? Okay, so the sort of punchline of this story is what's highlighted here. And actually, I should say one thing too. I know that students often get worried when I post a bunch of primary literature papers on D2L. Those papers are there as supplementary material to help you understand what I summarize in the PowerPoint slides. So if you want to know more about the study, you can dive deeper into that paper. But the key points I want you to know are always the things that I have posted on the PowerPoint. But that said, one of the goals I have for this course is for you to be able to critically read the scientific literature, because you will be preparing your own manuscript as your ultimate final project for the lab component of the course. So I encourage you, highly encourage you to read the papers that I post and have a look at them and think about the things that we've talked about other than just today's exact lecture. Like this paper has a bunch of great statistical outcomes that are reported. So look at those stats, and they have their sample sizes are reported. They've got a bunch of figures and tables in the paper. Have a look at those and see if they make sense, and do they stand alone, like we talked about in the first lab? Can you interpret that figure just by reading the caption of the figure and understand what it means? So think critically. Always think critically as you read them. Okay, so this story, again, is about these two mosquito larvae. So two mosquito species that have dramatically different saline tolerances on their curve, right? So we have a freshwater species and that euryhaline species. But what's interesting is that they have very similar pH tolerances, so they can handle similar acidities. And what's really fascinating is that mosquito larvae, as it turns out, seem to be one of the most pH insensitive groups of animals in existence. So that's quite fascinating. Both species are able to complete their larval development in waters that ranged in pHs from really acidic 4 all the way up to quite basic pH 11, which is really interesting, right? So we talk about the fitness response. So here we're talking about completing their larval development, which is obviously a key component of survival and going on to become an adult to go then reproduce themselves. So what's interesting is that both species can do their larval development in this wide range of pHs. But in a homeostasis type of response to this variable pH in their environment, they're able to maintain a very narrow pH in their internal body, their hemolymph, right, which is the blood of an insect. They're able to maintain a pretty narrow range of pHs internally, despite this really wide range of pHs externally. So that's pretty interesting in terms of a homeostasis aspect of this. And what's fascinating is that if you do an instantaneous transfer from one extreme of the tolerable pH range to another, it had very little effect on the growth and development of the larvae, which is also really interesting because I told you the example of a fish who has a shifting tolerance range to temperature in summer ponds versus winter ponds. But here's an example of an organism that doesn't seem to have that restriction. It's pretty good at tolerating this wide range of pHs. And so that's quite fascinating. And they did some further experiments with the freshwater larval species. And if you acclimate it to these pHs, to that range of pHs, it actually increases its survival when you push the envelope even further to pH 3, like really acidic, and pH 12. So if you acclimate those larvae to pH 4, you can get them to survive better in pH 3. Again, you can push those limits, showing that there is this acclimation process still happening in these organisms. So I thought that was a really fascinating paper. And it's published in a really good journal. Journal of Experimental Biology is a really good outlet. And if you look at the paper, in the introduction, they're using, like I said, the key words that we've been talking about. They talk about homeostasis. The pH of the environment is a major physical factor that limits the distribution of organisms in aquatic environments. Those are the exact things that we're talking about. It talks about larval mosquitoes can tolerate these ranges of ambient pH that are much greater than those tolerated by other aquatic animals. So they're these pretty incredible organisms. And so the purpose of the study was to investigate the effects of this ambient pH on the growth and development of larval mosquitoes. And they use larval mosquitoes that have different tolerances when it comes to their saltwater tolerance. So again, a pretty cool study. And so have a look at it. I can't remember if the version I posted on D2L has my highlighting in it already. I think it does. So that gives you an idea about what I was looking at to create this summary of the parts that I think are sort of the most important components of that paper. But again, it was really interesting paper that fits so well with what we've been talking about. Okay. So a little bit more about homeostasis, but now we're going to specifically talk a little bit about temperature homeostasis. So that paper talked about pHs and salinities. Now let's talk a little bit about temperature. So for homeotherms, right? Homeo means same, therm is temperature. So that means constant body temperature, right? So maintenance of homeostasis, thermal homeostasis, is mostly physiological, but it's also aided by some morphological and behavioral things, right? So, for example, in birds, which are homeotherms, to avoid summer heat, they might seek a shady spot, and they might use a behavior. So there's, behavioral. They'll go find a shady spot, or they might use another behavioral mechanism that's called gular flutter, which is the flutter of their throat, and that does evaporative cooling so that they can cool off. And then in winter, they can do things like added insulating down feathers, right, through physiological responses to the changing photoperiod. So there's a hormonal response that increases the growth of down feathers to keep them warmer in the winter. Okay, so a homeotherm then is an organism like a mammal or a bird that has a body temperature that's constant. Homeo means same, a temperature that's constant, and it's largely independent of the temperature of the surroundings. And so typically here we're talking about endotherms, okay? So we're talking about organisms whose heat is generated within, endotherm. Heat is generated within. So this is metabolism. They generate their heat through metabolism, which is generated then from eating to fuel that metabolic furnace. I just saw a chat, so I'm going to have a look at that. Would migration be another example? Yes, migration would be another example. Go seek a better place. Yep, that's a good question. Okay, so again, these homeotherms tend to be endotherms, but I'm going to come back to that. And remembering that an endotherm, endo means from within. So that means heat is generated from within. And in this case, it's through metabolism, which means feeding to fuel that metabolic furnace. Okay, and then poikilotherms. This means that they maintain temperature homeostasis mostly by behavioral mechanisms. So they must gain heat from or lose heat to the environment. So they'll bask to warm up, adjust body position so it faces the sun to warm up, and then retreat to a cool, shady place, go in a retreat to cool off. And so again, poikilotherms are using mostly behavioral, but they can also use some physiological mechanisms for some aspects of their homeostasis. So maybe not temperature, but maybe osmoregulation. They will be using then some physiological mechanisms for homeostasis around those other attributes. Okay, so poikilotherm is an organism like a fish or an amphibian or a reptile that has a body temperature that varies with the temperature of its surroundings. Okay, so these tend to be ectotherms. They get their heat from external sources. Ecto means outside, therm, temperature, right? So heat from external sources. Now, I want you to keep in mind, though, that many ectotherms, of course, are poikilothermic. Their temperature varies. But you can have some ectotherms that are pretty much homeothermic. There are some ectotherms that are really good at using behavioral mechanisms to regulate a fairly constant internal body temperature, right? So one thing could be through behavior. They could be choosing microsites that keep their body temperature fairly stable, right? So that's one aspect. Another could be maybe it's an ectotherm that lives in an equatorial region. So it has a fairly constant body temperature because it lives in an environment that has a fairly constant environmental temperature, right? So keep that in mind that many ectotherms can also be homeothermic if they're really good at regulating their body temperature. So that is to say that homeotherm and endotherm are not the same term, and poikilotherm and ectotherm are not necessarily the same term. Okay, there's slight differences to the definitions, and to keep in mind that an ectotherm can successfully be a homeotherm. Okay, sorry, I lost my advance. There we go. So another aspect that's related, but it's not totally related to the story we're talking about, but I want to bring it up just to bring it up. So this idea of autotrophs versus heterotrophs is just an important context to set. So, all life on Earth is composed of carbon-based compounds, right? And the acquisition of carbon might be the most fundamental process for life. So To assimilate new tissues, to meet metabolic costs, and to produce offspring requires getting carbon, right? That's essential. And not all organisms get their carbon in the same way. They don't all get it from the same source. And so one of the main dichotomies, two-way splits in ecology is this distinction between autotrophs and heterotrophs. Okay, so autotrophs, these are organisms that utilize inorganic sources of carbon, like carbon dioxide. So they fix carbon dioxide into organic compounds using energy from the sun, right? So these are the primary producers. This is some plants and algae and some bacteria, okay? So these autotrophs, these primary producers that use inorganic sources of carbon. So they fix carbon dioxide into organic compounds, and they use energy from the sun to do that, which is incredible. And then there's heterotrophs, and these are organisms that use organic sources of carbon. So they consume other organisms or their byproducts, and so we call them secondary producers, right? So this is animals, some bacteria, some fungi, okay? And so again, secondary producers. And then we can further divide those up, right? Consumers feed on other organisms, either living or dead, and then decomposers that feed on dead organic matter and waste products, okay? So the different ways that organisms get their source of carbon is going to impose different sets of constraints that directly influence how they interact with their environment, right? So this relates back to this idea of tolerances of different environmental conditions, because the different ways that animals are going to get their source of carbon is going to impose, different sets of constraints that influence how they interact with that environment, right? So that's how this sort of relates back to this idea of tolerances. Okay. So now I want to sort of use an example to talk about a life history trait and adaptations. Okay. It is 5 to 11, so we're good. Okay. So this is my adaptive story about clutch size in birds. So we're going to take a look at some of the evolutionary aspects around this. Okay. So, asking the question, what is going to determine clutch size in birds? So thinking about this. So each year, emperor penguins, they lay one egg. That's it. They only ever lay one egg a year. Pigeons, well, they can lay 1 or 2 eggs. Gulls typically lay 3 eggs. Merganser ducks, they can lay 9, 10, or even 11 eggs on average. So they have much larger clutch sizes. And so there's, what determines this? Why do those emperor penguins only lay 1 egg, and why do mergansers lay 9 or 10 eggs? And so there's 2 different aspects to consider in this question about what determines the clutch size in birds. And now we talk about that, the words I alluded to earlier, proximate and ultimate factors. So the proximate factors, so these explain how a trait is regulated by an individual, right? So this is, for clutch size, this is going to be related to factors like the physiological factors that control ovulation and control egg laying, okay? So proximate factors have to do with how an individual bird is decoding its genetic information about egg laying, okay? So clutch size might be modified by spring weather, population density that's already there, habitat suitability, right? So these proximate factors, again, this is how the trait is regulated by the individual, how that individual decodes its genetic information about egg laying in this particular case. Okay, so clutch size can be modified by weather, population density, and things that the bird detects around it. But then there's the ultimate factors, and these are the selective forces, right? This has to do with evolution and natural selection. So the ultimate factors that determine clutch size are the requirements from a long-term evolutionary survival perspective. So ultimate factors are always selective factors, and ultimate explanations for clutch size differences always involve evolutionary arguments about adaptations, right? So we're talking about evolutionary arguments about adaptation. So ultimate factors have to do with changes in the genetic program. So remember, proximate factors had to do with how an organism decodes its genetic information. But for ultimate factors, we're talking about changes in the genetic program through time and the reasons for those changes. So clutch size is an adaptation that's under the control of natural selection. So we're going to talk about those ultimate factors, right? That's our discussion today is about ultimate factors. So we're going to just focus our discussion on that. So natural selection is going to favor the birds that leave the most descendants, right, to future generations, right? Your whole goal in life is to survive and reproduce and leave your genes to future generations. That's the motivational force for life. Okay. So natural selection should favor the birds that leave the most descendants. So it seems pretty logical then to hypothesize that natural selection is going to favor the clutch size that's the physiological maximum that a bird can produce, right? Because you want to maximize your genes in the next generation. So then you should be producing as many eggs as you possibly can to do that, right? Okay, so we can test that hypothesis, right? Natural selection should favor a clutch size that's the physiological maximum a bird can lay. We can test this hypothesis by taking eggs away from nests as birds lay them. So birds, they lay their eggs in a nest, and then you do an experiment where you take an egg out every time a bird puts one in. And so when this is done, we find two results. We find that some birds, like pigeons, for example, they lay a certain number of eggs no matter what. So if you take an egg away, they don't lay another one. They have a fixed clutch size, okay? So we find that we have some species that are determinate layers. Again, so like this pigeon here. If you take an egg away, she won't lay another one. There's a fixed clutch size. It's not variable, okay? Determinate layer. But for other birds, they're indeterminate layers. They'll continue to produce an egg every time you take one away until the nest, till the nest is full. So if that bird, if it laid 5 eggs, you take one away, it'll lay another egg to get back to 5. If you take it away again, it'll lay another egg to get back to 5. It tries to maintain that number. So if the eggs are removed after they're laid, the female will continue to lay more eggs to replace the ones that are removed. In such a removal experiment that was done, a female mallard duck laid an egg per day until she laid 100 eggs. The same has been shown, for example, herring gulls in an experiment. A female kept going until she produced 16 eggs, but usually she'd only lay 2 to 3 eggs. Another example is the flicker. They usually lay 6 to 8 eggs, but they convinced a female to lay 71 eggs by constantly taking away an egg. And a house sparrow, they usually lay, little tiny house sparrows, they lay 3 to 5 eggs in the average clutch. The researchers were able to get a female to lay 50 eggs when they kept taking, like, holy moly, that's a lot of eggs that she kept producing. So we have these indeterminate layers. So this evidence, this indeterminate laying attribute, this indicates that most birds under normal circumstances, are not laying their physiological maximum. It's not supporting that hypothesis, right? This evidence of these indeterminate layers shows that most birds under normal circumstances aren't laying their physiological limit that they can handle. That female house sparrow can lay 50 eggs. So why doesn't she do that, right? Wouldn't birds want to leave as many offspring as possible? Why doesn't she lay 50 eggs every time? So it was found that most birds don't lay this limit, and that ovulation instead is stopped long before the limit is reached. So then that begs the question, why? How do you explain that? So a question here, but wouldn't the extra eggs be unfertilized? There wouldn't be any offspring. Okay, so they may or may not have been fertilized. So females will store sperm, and so they fertilize the eggs as they're coming out the oviduct. So they probably were fertilized, I would assume. But that's sort of a small piece of the story. The piece of the story that's relevant at this moment is the fact that she kept laying more eggs, regardless of whether or not they're fertilized. So what that experiment showed is just that females were not reaching their physiological maximum under normal circumstances. Okay, so we'll come back to that. Good question. Okay, so we come to this idea about clutch size as a life history trait. So I want to briefly talk about this idea of life history traits. So easy for me to say. Life history traits have to do with survival and reproduction, right? So they have to do with fitness. And so just to sort of give a list of what these are. So this British ornithologist, David Lack, he was one of the first ecologists to look at these life history adaptations, these traits that have to do with survival and reproduction. So examples of life history traits, the age and size at maturity. So when an animal, what age and size it's at when it first reproduces. Longevity. Reproductive frequency. Does it reproduce once a year, twice a year, 3 times a year, once in its life, or multiple times over its life? Reproductive output, so clutch size. This is what we're talking about today. We're talking about this life history attribute. Also, age-specific fecundity and age-specific survival are life history traits. Young individuals might have a higher survival than older individuals, or vice versa, but that's what that's talking about, okay? So again, the life history trait we're interested in today's discussion is reproductive output. Okay, so in 1947, just gonna check the time, we're good. In 1947, Lack hypothesized that clutch size in birds was determined ultimately by the number of young that parents can provide with food. So this is an explanation for why those indeterminate layers weren't laying 50 eggs at a time, because who can take care of 50 kids at once, right? So the hypothesis that clutch size in birds is determined, this is an ultimate factor, by the number of young that parents can provision. So we're talking about an animal that has parental care, right? So this sort of applies directly to birds. And so if the idea is correct, then this explanation, this hypothesis, if this is the right explanation, then the total production of young should be highest at the normal clutch size that we observe, right? So for those house sparrows, that 3 to 5 egg clutch size, even though that female can lay 50, that we should see the total production of young is the best at that clutch size that we tend to normally observe. And that if you experimentally increased clutch size by adding more babies to the nest, then you should see greater losses overall because the parents won't be able to feed everybody, right? So that's the predictions, those are the predictions that you would expect to see, right? So again, if this hypothesis is the right explanation, then the total production of young should be highest at that normal clutch size we see, because if you add extra babies to that clutch, if you experimentally add extras, that increased clutch size should result in more losses of babies, unsuccessful babies, because the parents can't have the provisioning for everybody. So here's a theoretical model of looking at that. So one way to think about this sort of optimal clutch size idea is to think about it in an economic approach. So everything an animal does is going to have costs and benefits, right? And an animal is going to integrate that information about costs and benefits over evolutionary time. And it should do things that maximize benefits over costs, right? And in this economical theoretical framework. So the benefits of laying more eggs is really clear, right? Because you're going to have more offspring, more descendants in future generations. So you're going to have more genes in future generations. The costs are a little bit harder to talk about, but we're going to talk about those. So, you think about it in terms of animals have a finite amount of energy, right? They have a pie and they can slice it in different ways, but there's a finite amount. So there's an energy cost to make each additional egg, and then there's cost to feeding those additional nestlings, those additional babies. So if the adults have to work harder to feed an extra baby, there might be costs on them for future reproduction. They might not survive because they're so exhausted from taking care of these extra kids. And if there's too many nestlings, nobody will get enough and everybody might die, right? The parents just can't feed everybody, right? So too many babies for the parents to take care of. So here's a cost-benefit analysis of how that might look. So on the x-axis, we have clutch size, and on the y-axis, we have units of fitness, and we have a benefit curve, and we have a cost curve. So again, of course, the benefits, right, is more offspring and more of your genes in future generations. And then we have all those costs that I alluded to, and we'll talk more about on the next slide. But if you look at under this theoretical context, the maximal difference between those costs and benefits, that would be the optimal clutch size, right? Where you're maximizing the benefits. And in this theoretical example, that would be a clutch size of 4 eggs, right? So again, clutch size with the maximum difference between benefits and costs is the optimal clutch size. And because this is research that Lack did, this optimal clutch size has been called the Lack clutch, right? It's been given that name, the Lack clutch, named after David Lack for the research he did on this life history trait on clutch size, okay? So again, this is the theoretical model that, this is how you would predict it. So let's apply this to some real data, okay? So here's a test of that theoretical model. So Lack's hypothesis indicates that clutch size is under stabilizing selection. If you remember when we talked about types of natural selection before. So can somebody tell me in the chat what would stabilizing selection, how would that look? If you look at this graphic for example, on clutch size, what is stabilizing selection going to do? It's going to narrow the curve. That's right, Cameron. The birds would get closer and closer to laying 11 eggs per year. Right. So in that stabilizing selection context, right, the lower clutch size and the higher clutch size are going to be the ones that are culled from the curve, basically, until you get narrow and narrow, centraling around that. Sorry, that was my alarm. Centraling around that 11 eggs, right? So it's under stabilizing selection. Thank you for the replies there. So individual birds appear to produce the clutch size that maximizes their reproductive potential. And this clutch size is smaller than that physiological maximum. And so again, this optimal clutch size is called the Lack clutch. Optimal clutch size equals the Lack clutch size in respect of David Lack's research on this topic. So that means that birds produce, on average, clutch sizes that are smaller than that maximum that's possible, that physiological maximum. So we ask why. And this goes back to the defining what those costs are, right? So here they are. So clutch size may affect the chances of the adult bird surviving to breed again, right? So adult birds are vulnerable to predation when they're taking care of their kids and they're incubating those eggs and caring for the young. So it might have an impact on the chances of the adults surviving to breed again. Also, there's this idea that I mentioned already about exhaustion from rearing large clutches. When you're taking care of a whole bunch of babies and feeding them, that's a lot of work. I watched a robin family, 2 robin clutches evolve on the kayak hanging from the roof of my carport this summer. And man, that female was back and forth constantly feeding those babies, especially when the babies were getting bigger, right? So it's a lot of work. So birds might become exhausted from rearing large clutches. And that's energy invested now means less energy for later for that female. Laying large clutches might postpone the next breeding event because it was so expensive, right? And so that means a reduction in the overall lifetime reproductive success, okay? And then also large clutches just themselves, right? It's energetically expensive. Eggs are really expensive things to produce. So that means there's less energy again for future clutches. So it's in this context again of trade-offs. What you invest now means less energy for later, right? Your survival now is important if you want to survive later to keep reproducing, right? So that's where this idea comes from about those costs, okay? So if we look at this example here, so this is an example of the production of young by blue tits in England at Windham Wood. This is actually a really famous long-term study site for these birds. So they used females that had laid 11 eggs in the previous year, right? That's who's used in this experiment. Because we expect those individuals are going to have their highest fitness at that 11 eggs, because that's what was observed for them the previous year again, right? Under that hypothesis that what they produce is what is the optimal clutch size. So these results here fit the Lack hypothesis because adding more chicks and just after hatching doesn't increase fitness. So here's the experiment. So when chicks were added, right? So again, for these females the previous year, they produced 11 eggs successfully. So that's assumed to be their optimal. So now we're testing it. So then when you add extra babies, they actually had a reduction in survival. So the y-axis here is offspring survival. So when you add chicks, there's a reduction in that survival. And then on the flip side here, if you remove chicks, well, you have fewer offspring surviving because there's fewer offspring there anyhow, right? So, but this experimental approach of removing chicks and adding chicks confirm the hypothesis that 11 is the optimal clutch size that parents are able to take care of, okay? So I hope that makes sense. Okay. Okay, so just a bit of a recap on the things that we talked about today. Adaptations, right? So any heritable phenotypic trait that there is the result of natural selection, and it's going to enhance survival and reproduction. So remember, fitness is that survival and reproduction is, and it's relative to other individuals that you're competing with in your population. So that law of tolerance we talked about. So this is about the environmental limitations on reproduction, growth, and survival, right? That bell-shaped curve. Organisms can survive, and they have a fitness curve within that range of environmental conditions, whatever they might be, temperature, salinity, etc. We talked about wide ranges and narrow ranges. And then we talked about homeostasis, that maintenance of a constant internal condition versus a variation in the external conditions of the body. Talked about endotherms and ectotherms. Endotherms get their heat from inside when we talked about temperature homeostasis, versus ectotherms that get their heat from external of the body. And then homeo versus poikilotherm. Homeo means constant temperature. Poikilo means variation in temperature. Okay. And then we finished by talking about an adaptive story, talking about a life history trait, clutch size. We talked about the work of David Lack to test the hypothesis that birds are producing the clutch size that produces the number of babies that they're able to successfully take care of, right? So that optimal clutch size is not the same as that physiological maximum. And the reasons have to do with this cost-benefit analysis that hinges on the idea that females produce, families of male and female birds produce the number of babies that they're able to take care of and successfully fledge. And that is today's lecture. And I will stop sharing screen and go back to the chat to see if there are some other questions that maybe have come up. We have about 5 minutes. Okay, so there was a question earlier. Do they maintain the number for the number of chicks they can support? Yes, so that's the lack, I think that's what Janine is asking. So that has to do with the lack clutch, the optimal clutch size, and that has to do with the number of babies that they can take care of, exactly. And then Cheyenne asked, why don't the birds that are the victims of nest parasites lay more eggs each time its own egg, I think, is pushed out of the nest? Would it be too late by the time the parasite hatches? Well, that's a fascinating question, actually. So these brood parasites, right? These cowbirds and other brood parasites that come and put their eggs in somebody else's nest. And so in that case, I think that it's that the stimulus is different, because at this point now you have the baby of the parasite, the brood parasite bird, is massive, and so it's giving the supernormal stimulus for the mother of the parasitized species to keep feeding it. So if you take animal behavior, when I took animal behavior a million years ago, there was this concept of a supernormal stimulus. When a baby bird opens its mouth, there's a color pattern in there, and it stimulates the mom to want to feed. And so I think when it's, my hypothesis about this would be that once they're at that stage, the mom of the parasitized species, she's not thinking about laying eggs anymore. She's just thinking about feeding that baby because of the supernormal stimulus. So I hope that sort of answers your question, Cheyenne. And again, I'm just kind of speculating on what I think could be happening. Okay. Okay, are there any other questions or comments in the last couple of minutes of the lecture? I'll hang on for a minute to see if there is. And you're free to go if you are ready to go. Some questions about the lab. Okay, Kiana, do you want to ask your question about the lab? You can type it, or you can turn on your microphone if you want. Assuming you can turn on your microphone if you want, Kiana. My microphone doesn't work on my computer. Well, okay, she'll type it. Yeah, actually, and if you guys, my camera on my computer is just totally worn out, so I always look really yellow. But I have a new computer coming. Hopefully it has a better camera than this worn out one. I'm not that jaundiced. It's just the really horrible camera on my laptop. Okay.
I don't know if you had time to look at your emails yet, but I sent you one yesterday about the lab.
Yeah, Caroline, I did see that. I haven't had a chance to answer it yet,
Okay.
but I'm going to check with the TA. So Caroline's question was about for the bean lab coming up next week, whether or not each of you needs to bring all of the implements for collection of those beans. And I'm going to say yes at the outset, but I will consult, because I want you guys to each get the experience of doing the different, at least have a couple of different implements for collecting your beans. But I think the idea is that your lab section everybody will be doing some different bean collection tools. And, some of you may do more than one type, and then all the data will then be gathered into one set for your lab section. So I think that's going to be the answer, but I'm going to consult with the TAs, and I have that on my to-do list for this afternoon.
Okay, thank you.
You're welcome. Yeah, and so you've all also related to that have been now assigned to your lab sections. I used D2L to assign you, and then I sent you all emails through D2L, which I've never done before, but I hope it worked out so that you now know who your GTA is. And I'm still working with the TAs on, so GTA means graduate teaching assistant, TAT, so it's the same thing. It's just that they're grad students. That's where the GTA comes from in that acronym. But I'm going to check with them. We're to try to decide on how we're going to do the pre-lab talk, whether one GTA gives the pre-lab to everybody and then you all go into breakout rooms with your individual GTAs to do the exercise, or whether your individual GTAs will do an individual pre-lab with you. So we're still working out how the TAs want to do that. I want the labs are, I mean, I've written the labs and provided all the info to the TAs, but they are the ones running the lab. So related to that, if you have specific questions about the lab, direct those to your TA. You can copy me on your questions, but for the most part, they're going to be the ones that are, for the most part, in charge of how the lab goes. And again, yeah, copy me on it because this is the first time we're doing this, so we're going to see how it all goes. Okay, so Kiana asked, I joined the class this week and I have missed the last few weeks in terms of the labs and classes. I've gone through all the missed slides and looked at the labs. In terms of lab quizzes and all that, is there anything I may have missed that's crucial to grading and all that? So no. So at this point, Kiana, you haven't missed anything that's crucial. Everything is posted on D2L that you need to know. The first lab was me giving a lecture on scientific writing, and all the info that I talked about is posted in D2L so you can get it. But you can also talk to other students in the lab, in the course, and you can also talk to your GTA when you go to your first lab on this coming Monday about any details. But all the lecture content and lab content has been posted. The syllabuses are there also on D2L, so have a look at those for where you're going to be getting marks in the course. Okay. And now we are past our course time, so I will say goodbye to everybody and wish you all a good weekend. And I will close down the lecture.